Transcription factors are like orchestra conductors. They turn on and off our genes. But in order to work, they must recognize the specific sequence motifs in the genome so that they can bind with high affinity and perform the function change in the expression of a gene nearby. We have already talked about some transcription factors in previous units. They have very important roles in development, in morphogenesis. They control how different body structures are formed, so it is quite reasonable to expect that they will also be involved in evolutionary changes. But how can we know? Well, at the end of the year 2013, an international consortium of scientists analyzing the human genome in great detail made a staggering discovery that intergenic regions with no genes that link one gene with the next, part of the so-called dark matter of the DNA, are full with short stretches of sequence that act like switches. They are regulatory elements, that is, they act as binding sites for many different transcription factors. And when I say that our genome is full of these elements, I'm not exaggerating, because there are several millions of them. As the sequence of the genomes of more species became available in recent years, it has been possible to study to what extent non-coding regulatory elements and sequences have been instrumental in bringing about evolution of form and function. One pioneer study, published in 2011, identified some 3 million of these short DNA motifs that were very well conserved during the evolution of vertebrates. That means that they are found in the genomes of most modern vertebrates, from fish to human. Then the authors of that study did two very interesting things. First, they looked at the functions of the genes regulated by each of these switches. That's fairly straightforward. But looking at the rate at which those sequences mutated during evolution, and this is the most extraordinary part of this study in my opinion, they could date the moment in which each sequence began to work as a regulatory region, because the mutation rate slows down when a region becomes functional. And what is remarkable is that apparently all these regulatory sites became active in 3 successive waves over the last 500 million years. And the switches activated in each of those waves regulated specific types of genes and proteins. For instance, the first wave dates back to 500 million years ago and involves genes active during embryonic development, genes that code for transcription factors. And this fits in quite well with the diversification of the repertoire of transcription factors that took place at the origin of the vertebrate radiation about that time. Then a second period, around 300 to 200 million years ago, saw the birth of many regulatory sites, preferentially in genes encoding receptors. And the switches that were activated more recently, in evolutionary terms, that's in the last 100 million years or so, were enriched near genes responsible for the post-translational modification of proteins. So what I find really incredible is that we can sort of reconstruct macroevolutionary trends just by comparing the genomes of distantly related species. But what this work highlights as well, together with later similar studies, is the importance of non-coding DNA in evolution. For decades, we had been focusing on coding DNA, the part of the genome that actually codes for proteins. But it might very well be that it is non-coding regulatory DNA that matters most in terms of evolutionary change. In fact, we know that these switches determine when and where many developmental genes are expressed. And developmental genes are crucial for the formation of structures and organs in the embryo. So let me illustrate this point with 2 very nice examples. One of them is very well known and involves our best friend, the dog. There are many breeds of dogs with short legs. Probably the best known is the famous Dachshund. This remarkable phenotype resembles in part what happens in a human genetic disorder called achondroplasia, a frequent cause of short stature due to the increased activity of a protein called FGFR3 in the growth plate of long bones. So the plate closes prematurely and long bones stop growing. When scientists analyzed the genomes of breeds of dogs with short legs and compared them with the genomes of dogs with legs of normal size, they found that Dachshunds and other short-legged breeds had an extra copy of a gene called FGFR4, a very close relative of human FGFR3. And this extra copy in dogs, which came from a retrotransposition event, had some changes in its regulatory region so that it was active ectopically. Ectopically means that it was active in a tissue where it should not be active. And that tissue, of course, was the growth plate of the long bones of these dogs. Another more recent and probably more impressive example of the morphological changes triggered by regulatory elements comes from the analysis of a HOX gene in the genome of a fish. All land animals with 4 limbs, known as tetrapods, share an ancestor that left the sea and invaded the land. This ancestor was a fish that could breathe in the open air and had a special type of fins with bones inside, like the modern lungfish. But most types of fish have the typical fins with rays. So how could this change have come about? The transition from ray fins to lobe fins. Well, as it happens in the embryo, both fins and limbs start with a very similar structure, a sort of bud. And in fishes, this structure has a large fin fold, as it is called, whereas in the tetrapod ancestor and the tetrapods, this fin fold is very reduced and is replaced by a region of cartilage that will eventually form a bone. Scientists knew for some time that genes belonging to the HOX-D cluster of HOX genes were very important for fin and limb development, and there are some differences in the way in which these genes are expressed in this structure in fish and in primitive tetrapods like the coelacanth or the lungfish. So a study published at the end of 2012 reported that what happens when you change the expression of HOXD13, one of the HOXD genes, in zebrafish embryos. Zebrafish is a ray-finned fish. When the activity of this gene was increased above normal levels, at the time when fins are starting to develop, they observed a reduction in the size of the fin fold and the formation of more chondrogenic tissue, cartilage. Exactly the type of changes that you need in order to make the limb of a tetrapod. And remarkably, when the researchers put the HOXD13 gene of the zebrafish under the control of regulatory elements, one regulatory element taken from the mouse HOXD cluster, an element that controls the expression of these HOXD genes in the mouse, the result was again an increase in HOXD13 activity and a change from the fin to the limb type of growth. Of course, a lot of work is still needed to elucidate all the details of this process, but incredible as it sounds, it might very well be that a few subtle changes in regulatory elements, in regulatory regions controlling the expression of one or a very few HOX genes, were in fact responsible for a major macroevolutionary event like the transition from fins in fish to limbs in tetrapods.